So good evening, everyone.
Good evening, sir.
Yeah. So today first we will discuss about some points like we did the analysis for genome-wide association study by using one genotypic data file, one phenotypic data file. Then we did GWAS analysis using all five models. Day before yesterday we did using single model, that is farm CPU. Then we sorted the significant markers. So before repeating that thing, first we'll discuss now that genotypic file we use. This file is called as HapMap file. So what is mean by HapMap file? So that HapMap file is one file format used to store the genetic data, particularly the single nucleotide polymorphisms. So if you will see, yeah, so this file is called as HapMap file. That's why the name is that MDP underscore genotype underscore test. So that is the format dot HMP. Basically, it is a TXT file. Dot HMP means it is a HapMap file, and it is extracted from haplotype map. So haplotype map means haplotypes are the sets of DNA variations. Sets of DNA variations that are inherited together on same chromosome. So chromosome wise sets of DNA variations like the single nucleotide polymorphism. So that sets are called as haplotypes. So from those haplotype maps, this HapMap file will be extracted. So as we know, this is a text-based file which is having this accession number, then this marker ID, on which chromosome and base pair yesterday, day before yesterday, we have seen that. And this either A or T, which form, like here we have seen A means A, this C means C. If the expression is combinedly for both the alleles, that is G and T. So likewise, that combined effect is represented by one separate letter. That is K for GT, M for AC, W for A and T. So likewise, it is represented here. So this HapMap file is extracted by following one procedure, that is GBS, that is genotyping by sequencing. So this genotyping by, if you have gone through that PDF I have shared, so in that you will find one procedure of genotyping by sequencing. If you have not seen, now also you can see that. There are total 1, 2, 3, 4, 5, 6, 6, 7 different diagrams, right from collection of tissue to bioinformatical analysis of next generation sequencing data. So by following that procedure, That procedure is the protocol for genotyping by sequencing. For finding of these thousands of SNPs for for that particular trait. So this is the procedure. There are two main methods, that advantages, disadvantages, that diagrams, everything is there. And along with SNP, again one type of marker is there, that is DART. That is diversity array technology. So along with SNPs, nowadays this DART markers are also used for the mapping purpose. So then again we have seen one kind of map called linkage disequilibrium map. That is, in short, we used to call it as a LD heat map. So what is meant by linkage disequilibrium? So linkage disequilibrium is non-random association of alleles. So what is meant by non-random association? Yes, anyone? Non-random association of allele means what?
So that means alleles are linked.
Okay, so that means alleles are linked to that particular locus or that particular trait.
So particular chromosome we don't know. That is randomly associating with a particular trait. Which chromosome, which region, we don't know. So that is called random association.
Yeah. Then yesterday we saw, day before yesterday we got some 6-8 SNPs, but when we check in the database we got only one. So can anyone tell me the reason? Yes. In Jivas, after analysis, we used to get 10, 15 markers. But when we'll check in database,
So
that all
false
15
positive we call.
markers, what is meant by false positive?
Sir, I didn't do the Jivas, but I'm answering on the basis of QTL mapping. So I'm trying to answer. So when we map, before we make a make a linkage map, the with the like what what we found during the study, the SNPs. So maybe due to due to what we make on the linkage map, based on the position, it should be there, but due to due to like when we make the crossing and something is change its position and it's not on the linkage, but according to the linkage thing it should be on the map. So maybe due to crossing and something it's changed the place. That's why we are not getting that marker on the chromosome position. Something is it possible or not? Maybe I'm not.
Okay, any other answer?
So due to like phenotyping mistakes or error, improper phenotyping, it may detect some false reasons as an essence.
Yeah, anyone else?
Sir, I'm not sure is it correct, but it's just a guess. I think the chromosome which showed the on the that mapping, I think that was the associated with the marker. That's why it was like shown the on mapping.
Yes, anyone? Yeah, strong statistical association. Okay. So whether in that false positive, whether there may be any role of population structure? So in false positive, whether there is any role of population structure? In what way? If population structure is having any role, so in what way? Hmm? How population structure is somewhat responsible?
The number of population is more, the accuracy will be more.
Okay, population structure, yes, it will assign the kinship values. So after assigning values, what will happen?
Sir, the genetic relatedness among the individuals in a population prevent the true discovery of the markers.
Okay, right. Just once complete perform that analysis with Jivas, then we'll proceed further.
Excuse me, sir.
Yes.
Sir, whatsoever we have studied in Jivas, that was for one particular trait. So, what if we want to perform Jivas for more than one trait? In that case, how will we be proceeding for that?
Procedure is same.
So, we have to go for that individually?
Yeah.
Okay, sir.
So generally for how many characters generally Jivas we will do? Any idea now? You are searching many papers, you are reading, some are working professionals, some are doing the PhDs, MScs. So generally, how many traits we will consider? Yes. Whether it is like we did stability for 10 characters, so whether you have found any paper with 10 Jivas for 10 or 12 characters at a time in one research.
No, sir.
No, sir.
Why? General question I'm asking why.
It won't be possible to do the genotyping and basically phenotyping simultaneously for that huge population for two traits or three traits.
So generally 1 to 2 traits only maximum it is there.
So the problem is with the analysis part or with the taking phenotype?
So problem in molecular breeding is money.
Yes.
So institutes like, suppose institutes like ICRISAT in India, I'm saying ICRISAT or these ICR institutions, they can afford. But suppose if you will say like state agricultural universities, so it is a very big task for them. In means as far as resources are concerned, then money is concerned. So it is literally difficult for state agricultural universities.
And what was the answer for the false positive, the final? I didn't get it, so I just want to know.
So actually, see, if you will, if we will do the Jivas with germplasms, right? So
Yes.
each germplasm, their origins will be different.
Yes.
Yeah. So on the basis of that ancestral relatedness, that's why someone told the answer that is because of family relatedness, means ancestral relatedness of each and individual phenotype.
Okay.
After analysis, we'll find that association, but actually when we will go for that, you will not find that position.
Okay.
Okay. Just try to run this Jivas with farm CPU.
Okay, sir.
And tell me whether any error is coming.
Sir, can you also teach us how to prepare this HAP map?
So for that HAP map, it is not possible with our systems. So we need that.
Just theoretically, sir, like
That I can tell you, no problem.
like how to extract data from the papers, then like little example just with one.
Yeah, that I can tell you, no issue. Describe Jivas is because I am getting error. I don't know. Take only one model. Don't go for more than one.
Yes, sir, it is showing error.
Start. Now it is going. Now it is fine for me. Start again. Completed. Yesterday's files are there. Output files are there in your working directory.
Yes, sir.
Yes, sir.
Yeah,
Yes, sir.
I think it is giving problem in overwriting that existing file. For first attempt, I also got the error, but in second attempt, it is completed. Yeah. So next thing is, yeah, who will tell this probability and false discovery rate threshold? P value and FDR. Now say after completion of Jivas, where is that output file we got? We got output file for 3042 SNPs. So how to select the significant markers from this file? Yeah, madam, you are telling. Continue.
Sir, based on the significance of association for p-value 0.001 and fault discovery rate 0.05.
Okay. So now in our file we have 3042. So how that two criteria will work? That is whatever you are saying, probability and false discovery rate. So how that two criteria will work for us and it will give us significant SNPs. Yeah, anyone? So how p-value and FDR will select the significant markers? Low probability, strong association. Okay. And what about FDR? Whether that FDR will be applicable for all 3000 or what?
No, sir.
Then,
It will be, sir, applicable for the markers which have been found significant.
okay. So, firstly, our criteria is we need the markers. If you will see the output file, we have SNPs, then second column chromosome, third column position, fourth column p-value. P means probability. So what is the meaning of probability?
Sir, your R studio is open. We are unable to see the Excel file.
Yeah. So what is the meaning of probability?
Probability is chances of
Chances
occurring any event.
Okay. So here we need to find the significant markers firstly on the basis of probability value. So lower is the probability value, more is the association of that particular SNP with that particular trait. So we need markers with probability 0.001. Probability of markers having less than value 0.001. Suppose 10 markers we got. After that, to control the false positive rate, our false discovery rate should be less than 0.05. So means after probability test, whatever the markers are selected, we need only 5% from them. Not from whole file. Whatever suppose 10 markers are there, we need 5% from them. So for that reason, we will run the script. So just run that script. No need to change anything. Already we are in that same directory.
So, excuse me, sir, when we are giving the command for FDR, at that command we are putting inside the bracket like GWAS at the dollar sign and then p value, like those who lines, those markers, they are selected after screening by the p value, then we will apply FDR on
FDR.
that.
Yeah. So then after that, Excel. After completion of this, you will get the significant markers. In the same working directory, you will get the file. So first file is probability value less than 0.001. Then in second, with FDR less than 0.05. Yeah, so see here, after going through this probability test, we got 6 SNPs. But when we applied FDR, out of that 6, only 2 are there. So what is the next part after this? Yes.
We will go on
To
five.
confirm if
Cytosome we will go and check, sir.
Yeah. Where we'll check?
Phytosome.
phytosome.
Okay. As this is the sorghum data file, so we'll go to sorghum genome. Yeah, so that will be open. But what to do here?
-1000 plus 1000.
Why?
So since probability is 0.001.
We have not discussed the answer, but yesterday, but day before yesterday, you people search in phytosome. So from that, whether anyone got any idea why we are preparing that range?
The marker lies in between the genes, the length of gene is more.
Yeah, but we got exact, but we have exact marker position, na?
So it lies between that base pair value.
Excuse me, sir. Why we use 1,000 base
Yeah,
pair?
yeah. I'm coming to that only. I'm coming to that only. Because see, if you will open this phytosome or any ensemble or any other data resources, generally it will ask like starting point and end point. Okay, so one way is like you can write here what is that position. We'll copy it. So at both places you can write this same. This is one way and you can go. So directly you will get gene, right? Otherwise, you can make one range. I am taking 1,000, but it is not. You can just make a range. Even you can prepare for 50 base pair, 100 base pair, 500. That is not a problem, but not more. Means it is. It should not be more like 5,000 or 10,000 base pair. So we are for what purpose we are preparing the range? Like if any other markers or any other desirable markers are there nearby that gene or are nearby that identified marker on the same gene or beside that gene, if any other desirable genes are there. So to see that thing, just ideally 500 to 1,000 base pair range will do. Otherwise, directly you can keep the position at start and end, you will get the gene. If this marker is on gene, you will get the gene. So if you will zoom, if you lower down this zoom, so this is your gene. So on this gene, here that marker on 4th chromosome, that is with this position, it is located on this gene. Means this is not a false positive. If you will double click on that, today it is working well. Thank God. Yeah, so see here. After double clicking on that, so this is the gene ID. Then next it will give details, that is with genome assembly. This is phytosome genome ID 454. Then the location. Then this is the description. If you want to go detail into that description, you can go. That is called as system biology. Then the functional annotation of that particular gene based on the different database. So these are the different database and these are the annotations. So what is meant by annotation? What is the meaning of annotation? Hmm?
Representing something, assigning its function, gene function.
So annotation means first it is identification, then identifying. First we identify the location, then what are the functions of that particular gene within that genomic sequence. So this is the gene. We identified first the location. Then second, from this these details, we'll get to know the function of that gene. So this whole process is called as gene annotation. Identifying the position and function of genes. That is called as gene annotation. So here number of databases are there. Then here you will get the sequences like genomic sequence, transcript sequence, CDS, peptide. Then where is homology? Phytosome. So in your case, it is working.
No, sir.
No, sir.
Yes, sir, it's working for me.
Yes, sir.
So yeah, if you will right click that genome, that gene, you will get all these sequences. So as we are searching this gene, we will see this in some time. As we are searching this marker in sorghum 3.1.1. Now let's see at sorghum 5.1 whether it is there or not. Where is 5.1? 5.1. You see the same marker on assembly version 5.1. Go to fourth. Just copy that, direct that position at both the places, and we'll see. Yeah, so what is the result? It is there or not?
No, sir.
I don't know, 5 to zoom is not working for me right now.
Oh. For those working, just they can check on 5.1. So see, in 5.1 also, this marker position is there on the same gene. But the gene ID is different. Here you will get the gene ID. In 3.1, gene ID was different. I'll open it side by side. Function will be same. See, it is saying protein coding gene. Yeah, so here I will open 3.1.
Sir, how you are seeing the gene? So, from where?
राजी इधर zoom in out का बटन है.
No, sir. After that, how we are seeing that name of the gene?
So if after double clicking it is working, then it's fine. Otherwise, right click on that and click on view details.
Okay.
So side by side, I will I have open this. So say this is the gene ID in 3.1.1 and this is the gene ID in 5.1. So this 5.1 is recently released in 2023. And 3.1.1 was released in 2017. So this marker is available in both the assemblies. Though the gene ID is different, but it is available in both. So just now, that 10th chromosome, just you can check. In 5.1. Yeah, see. This in 5.1, again we will get this new gene ID. This is total, this functional information with this database. After that, here we got the protein homologs. So what is meant by homology? What is the meaning of word homology?
Same.
Yeah. So protein homologs means whatever the protein coded by this gene, so similar, the similarity of that in different species of same crop and in different crops. So here you can see sorghum bicolor, different species. As we are dealing with 5.1, it is showing 100% homology with 3.1.1. Then here you can see the number of species with similarity percentage. There are number of species right from 100 up to, I think it is up to 40, 20.
Excuse me, sir.
Ah,
So right now my phytosome is open. Can I know where you are seeing this file, sir? Where we can open this?
which one?
This page.
Double click on that gene.
Okay, sir. Okay.
So likewise here, total details you will get. Again, these sequences, as I told you. See, these are the genomic sequences. So all this peptide CDS, and directly here from here you can go for blasting NCBA. So total details. I think this will open. Yeah. So now both versions are there. So likewise, information you need to identify. So now we'll search that marker on chromosome 10 in 5.1. This 10th chromosome marker we will search in 5.1. So is it there?
So some reaction came, so like chemical reactions.
Where?
So like alpha 1 phosphatidyl 1D. It's now,
Where?
sir, after double clicking on gene.
So see, this is gene. I will double click it. Like this it will come. Where is alpha?
At very downside, sir. At bottom most. Sir, sir, in mine it is showing, sir. I'm sharing the screen first.
So just normal double click you need to do on this gene, not anywhere here. On
Okay,
this gene, simply double click it.
Sir,
Yes.
hello.
Yeah,
Sir,
yeah.
in the protein homolog table, what is the meaning of that score and similarity on what basis? Structure or what?
It is based on the function. Whatever the functions are there. See, this is the gene ID. So this is the definition of that gene. Okay. And these are all are the functional annotation based on this different database. So based on all this information in sorghum, so now we need to check whether this gene is having any kind of similar work in any other species of same crop or any other different crop. So this is the list of related crops where this gene is having the same action, maybe 10% similar, 20%, 30%, 50%, whatever it may be, but it is performing same function.
Sir, means protein product is same or different? Because gene sequence is same but transcript is different. So,
Transcript will different na? Now see, I am running R, you are running R, that Raj is running R, but we all now, how many members are there? We all 41 members will try to work on R in our convenient way.
yes, sir.
No problem. You are learning from me. You are taking notes from me. But you will never ever follow the 100% same procedures. What I am following or what I am telling. So some 1 or 2% will follow, but mostly what you people will do, you will do modification. So it is like I am the identified gene. Someone will show 100% similarity, someone will show 30%, someone will show 10%. It is like that. So see, we have identified in sorghum, but still it is having 98% similarity in saccharum. Then Z maize.
Sir, in case of other than 100% similarity, gene product will be same or different? Means that protein.
You can see here on very right side last column.
Define.
Yeah, so here as the similarity percentage will change, whether you are observing any change there?
Yes.
So see, Z maize, that 88.1% similarity. All this there's all those 7, 8 species, their function is same. Some somatic embryogenesis or something is there. So all these functions means now we are working on fertility restoration. So maybe this particular gene is responsible for some other function. Though this protein, one protein is there, but maybe that protein will be responsible for some other trait also, in some other crops. You can't say like we worked on pollen fertility. So these are saying 88% similarity. So in what you will say in Bajra also it is responsible for pollen fertility. It is not like that. Gene will be same, but maybe that protein will be responsible for other character also. Because you may find this same gene ID for other trait. We are just dealing with one small marker on this gene. That's it.
Yes, sir.
Excuse me, sir.
Yes, Akshay.
Sir, we have used 2 kb range in this. Sir, in 1.1, the gene is having some somatic embryogenesis related, but in 5.1, it is related with the disease-resistant protein. Sir, why this is different in both these cases?
So actually see, as you know, year-wise, day-wise, systems will go on improving. Okay. So we have started from Android version 2, 3, 4, 5, 6, some errors, so they improved, some errors, so they improved, some errors, so they improved. Now in Android 10, 12, 15, 17, it will grow evolving. So it is like same. So if you will see the different genome assemblies, you will find like from 1.1 to 5.1. So in 1.1, some sequences are there, but many gaps. So because of that gaps, you will find some different result. In 2.1, maybe 50% gaps covered. In 3.1, some gaps covered, but still gaps are there. But now this 5.1 is from 1 telomere to that is T to T sequence. What is mean by telomere?
Terminal side of chromosome.
Terminal end of the
So
chromosome.
whatever the information on this 5.1 to genome assembly, it contains from one terminal of chromosome to the another terminal. So total information is available on 5.1 as it is a recent one. So that's why there is a difference in 13311 and 5.1. If you will check this on 2.1, you will get another some difference, 21311, 5.1. Same like our network, we can't use 2G now. So this assembly also it is keeps on improving. So if you will see the homepage of this, so see recent there. If you will check this date, see total updated versions are there. So if you will check on old and new, you will get some differences. Yeah. Have you checked that 10th position on 5.1?
I didn't get anything, so I don't know if the server is not working or
No, server is working.
for me it's not working right now, I think.
Sometimes for 10th, it is not coming for in 5.1 also. It is coming only for 4th chromosome. So that means the rest are, they are not exist. We'll check that second one also once.
So for chromosome 10, it's coming, sir.
Gene is coming.
After zooming out, gene is coming.
Check the position. Maybe it is beside that position. Check the position of your SNP on 10th chromosome. Tenth chromosome marker is not coming, as that is false positive. So that's why yesterday I told we got output with 3042 markers for that one particular trait. Out of that 3042, we got 6. From 6 we got 2, and from 2 we got 1. Yeah, so just here you can see the genome assembly version based on the accessions. So here you can see the accession numbers. Different, this 1.1 version only, but with different accessions. So just you can see 2.1 with different accessions. But then after that, after 2.1, then they are releasing the single version only, 3.11 single version and then 5.1. Earlier they used to release the specific accession wise.
Sir, what's the difference in the accession wise or the single one?
So in accession wise means based on that particular accessions and the whatever that progenies are developed, or the complete genetic structure of that accession. That's why you will find many more entries of 1.1 here.
Yes, sir.
As that was the first version of assembly for sorghum, so you will find many versions.
Okay, sir. And these many versions are with respect to the different accession of germplasm.
Yeah, just you can read the names. Names are different.
Okay, sir. And when we get with a single file, what does it mean?
So now that single means these assembly, they have combined and they are working on one reference genome.
Oh, there is a kind of reference genome for for all they are trying to make it.
All. If you
Like
want to
you have
download,
a genome in rice, the same.
Yeah, same. In each and every crop now they are concentrating instead of releasing entry wise or accession wise. Now each and every crop is like reference only. You will not find accession wise.
Yes, when I am working with Rhabdopsis, I find the same thing. The earlier one
Yeah.
has the accession, like different germplasm kind of thing,
Yeah.
different, and what we are getting nowadays, that is with the just one reference.
Yeah, reference genome only. Because now if you will check in accession wise, so in each accession you will get some different results. So it is difficult to conclude.
Yeah, but we need some time, like when we have to do the functional study after identification with the jiva something, and we want to do the functional study. At that time, we need the single accession to
Yeah.
validate that thing.
So for that purpose, after identification, we used to go for validation purpose.
Yes.
Or suppose after that again we will go for RNA expression.
Yes.
So for that we need to study that particular accession.
Yes. At that time, we need the accession data as well.
Yeah. Apart from reference, we need to concentrate on that accession also. So if you want to download that, again, one thing I will show you, you can download that genome assembly. Anyone is there working on Linux? Yeah, out of 40 members, anyone is there working on Linux? No one? Linux, Ubuntu. Yeah, so in this say, this is home page. Go to this right side, click here, and click on the JGI data portal. I think it is having some. It can. So if you have account, then log in. Otherwise, you need to register. Let us see it. Yeah, so after login, like this, window will come. When we'll go to this data portal home, so here it will search like Suram. It's taking time. So from here you can download the all the reference genome. It's somewhere near, I think 7 or 8 GB something. Yeah, 8 GB. This accession wise, and I think combine is also there. So likewise, all are there. Everything you can download it. Number of pages and go for next. So likewise from here, you can download it, whichever is required. If anyone just now, like Madam told, for functional analysis, if you need, just you will get from here. But its file size is too large. So it's better to go for Wi-Fi. So apart from this identification of markers, so you can download the assemblies also. And from that you can, someone can go for system biology, that is protein analysis. If you want to design also markers, you can design by using different tools. So if anyone is interested in bioinformatics, so they can go. That will be a added advantage. So it is like for one particular trait. So this is total jivas by using farm CPU. That is one model. So here then we'll come to our graph, Manhattan plot. Yeah, so say this is the Manhattan plot for screen is visible. Okay. Yeah. So see, this is the Manhattan plot. This is the chromosome, and these are the p-values. So say the system-based threshold level is nearby 5. And just see that on 10th chromosome, one data point is there on the top. So that marker we identified, but could not locate on the chromosome. So here we got that 10th position marker on 10th chromosome. And similarly on 4th, here is that data point. So more the distance, strong is the association, that is the concept. But in that concept, that false positive will come. So out of these two, we got this one present on chromosome 4. So this plot is called as Manhattan plot. Simultaneously, I will open Q.
So can you please explain again this Manhattan
So
plot?
these are observed or expected p-value. Which are these?
Expected.
Huh? Expected or observed?
Expected.
Observed. Sir, expected.
Observed.
How expected? Why not observed? Hmm? So these are the values here we got. So these are these values. These are these values located on y-axis. So these are the observed values, not expected. And whatever the dots are there, color dots, each dot is one SNP marker. And on this x-axis, these are the numbers 1 to 10. So these are the number of chromosomes. And these are the distribution of markers based on these p-values. So see here, in Excel, finally we got two markers. One is located on chromosome 4 and another one is located on chromosome 10, which are having lowest p-value and FDR is less than 0.05. So on chromosome 4 and chromosome 10, lowest p-values is for this marker and this data point. So data points here are nothing but markers. So these two markers we identified from our statistical analysis. We identified that these two markers located at chromosome 4 and 10 respectively, they are having strong association with this pollen fertility trait in case of sorghum. This is identification. But when we went for annotation, only we got annotations for this marker present on 4th chromosome. And this data point is false positive. Once I will open QQ, so you will get to know this.
Sir, in general, I have one doubt.
Tell me.
So, how much criteria will follow log to log value? Log 5 or log 6 or log 3 or log 4?
Come again, question. You are asking
So,
about
in
threshold
general,
level?
yes, threshold level. How much threshold level will follow?
Just see this graph. For that only I open this QQ plot. So generally see, this is the threshold line given by the analysis. But we want threshold value based on our data. So for that, this quantile plot will help us. So here on x-axis, expected value, on y-axis, observed value. And this is the distribution of our data. So this is the normal distribution red line. So our data distribution is started and the point where our data points are started diverting from the normal distribution. So this is the point from where this data is started deviating. So that point we need to consider as a threshold level.
So,
What
my doubt
question you
is
are
that
asking?
my question is in this farm CPU QQ plot, the round circles are they are reached nearly 5. So, can we take the threshold value log value 5?
So what I am telling the point from where the data points started diverting. So that point you have to take as a threshold level.
So here log 3 is this, the 2.5 is the threshold value.
So approximately you can take above 3 and above, whatever the data points above 3. Or above,
Okay.
if you
Here
want to
the,
take exact, just take above 2.5. So whatever the data points are there above 3. So threshold value you need to decide based on this QQ plot. Or else you can follow the system generated threshold level also. But this based on your data, it will be very good for the outputs. So always for threshold level, just refer the QQ plot and check the point from where the data starts deviating from your normal distribution.
okay, sir.
Yeah. So that is Manhattan and QQ. Yeah. So the next one, the terminology is MAF. Yeah. So what is this MAF?
So minor allele frequencies
Inform.
or major?
Yeah. No. It is minimum allele frequency. That means minor. So what is meant by minimum allele frequency?
Between the alleles, the frequency.
Yeah. So it is the difference between the frequency of allele. Just give me a minute. Yeah. So allele frequency is the difference between the frequency of alleles. Yeah, and so one more question I want to ask: if you are working on any particular trade, so if in phenotyping data, if you will calculate the heritability and genetic variability parameters, so whether that heritability parameter will play any role in selection of character?
Sir, it should be high, sir. Then only selection will be efficient.
Yes, sir.
High means what?
More than 60%.
Superiority between the parents. Comparing with parents, we need
Your
to.
voice is not clear. Voice is not clear.
So comparing parents, we need to superiority. So heritability will help, sir, in that aspect.
Yeah, more means how much heritability? Minimum percentage of heritability.
60%, sir.
It should be 5% or 10% variation will be there.
Okay. So that whatever the traits we are selecting, so that heritability should be on the higher side. Somewhat 5-10% is fine, but that should be on the higher side. So then and then only the selection will be useful. So then yeah, so here this is This is like we got during our quantitative analysis. So these are the which plot we got during our quantitative analysis like this in case of PCA. What is the name of that plot?
Free plot.
Yeah. So here below the principal components and then the variance. So here in Excel you will see the eigenvalue for all the accessions. Right from below 1 to maximum, I think. Yeah, maximum is 81. So this is maximum. So then so this is the 2D plot of principal components. So as during analysis we mentioned first three components. So here all the three components are there. This is the once we'll run for all five. That's circular Manhattan event. Yeah.
Sir, hello, sir. Am I audible?
Yes. Yeah. Clearly.
Sir, today only I have joined, sir, this class. I need yesterday and Jiva's classes, sir. Can you provide?
Yeah. No problem. No problem. You will get. Don't worry.
How can I get, sir? Can I any recording mail will get or?
I will contact you through WhatsApp. No problem.
Okay, sir. Okay. Thank you.
Yeah, Bibakar sir.
जी सर.
हाँ जी. Over to you.
Thank you so much, sir. Dear participant, for the next 3 days there will be no session on the occasion of Ashtami, Navami, and Vijayadashmi. And after the Vijayadashmi, the next very day, your session will be start. As everyone want to enjoy their festival, so there should be a break. During this break, our office will be open, and in this during days, we are going to post all your recorded session. But there will no classes for you all. But we will be active for your work. The participant who have not joined any session, you will get all the recorded session in the given WhatsApp group. Don't worry. सभी को सूचित किया जाता है कि अगले 3 दिन तक अष्टमी, नवमी और विजयदशमी का सेशन नहीं रहेंगे आप लोग के लिए. विजयदशमी के अगले दिन से आप लोग का क्लास शुरू होगा और तब तक हम आप लोग का जितने भी रिकॉर्डेड सेशन है आपके व्हाट्सएप ग्रुप में, यूट्यूब चैनल पे हम लोग पोस्ट कर देंगे ताकि आप लोग देख सकें। आप लोग फेस्टिवल को एंजॉय कीजिए। तब तक हम आप लोग का सारे रिकॉर्डेड सेशन डाल देंगे ताकि आप लोग को किसी तरह की कोई समस्या ना हो। नाउ इफ एनीवन हैव एनी क्वेश्चन यू कैन आस्क। अगर किसी के पास कोई क्वेश्चन या प्रॉब्लम या सजेशन है तो आप बोल सकते हैं।
Yeah, and one more thing I need to say that regarding assignments, I am preparing data sets. So either by night or tomorrow morning, you will get the data sets. You need to analyze right from RBD, genetic variability parameters for a correlation path. For that, I will give you separate. For stability, I will give you separate. And then for this genome-wide association, I will give you one separate this HapMap file and phenotypic data. And for in that multi-location data, you need to go for box plot, heat map, chart diagram, everything. So means so far whatever we completed, for all those part, I will share you the different data sets. So you need to do the analysis, and like last time you shared with me, so like that you need to share. Yeah, so now if you have any questions or anything to say, you can.
Yes, the next session will be on October 3rd.
Video links where we will get?
Ma'am, that we are going to post tomorrow or day after tomorrow.
On
One by
WhatsApp?
one. On the YouTube channel it will be posted and the link I will share in the WhatsApp group.
Okay, sir. Thank you.
मैम छुट्टी आप लोग के लिए मैम, हम लोग के लिए नहीं है।
Yes, thank you, sir.
थैंक यू मैम। और किसी के पास कोई क्वेश्चन, कोई डाउट, कोई सजेशन है तो आप खुल के बोल सकते हैं, कोई दिक्कत नहीं है।
Sir, if we have any doubts, how can we contact sir? Just now teaching about partner.
Okay, regarding the topic, if you have any doubt, you can direct contact to Gopal sir. His number is there in the WhatsApp
Because
group.
many members I am in contact with, I think almost many members, they are sharing their difficulties in form of screenshots. I am replying whenever I am getting time. I am replying to everyone. But sometimes as I am also working 9 to 5, so sometimes for me also it is difficult to give reply on time. So maybe there may be delay in reply, but reply will come. Whenever I will get time, I will reply.
Excuse me, sir.
Yes.
Gopal sir, can we also get the interpretation of PCA plots and MEs?
Yeah. I am preparing everything, whatever we have completed. So I will share through your mail ID. You will receive the files on your mail ID.
Thank you, sir. Thank you so much.
And dear participant, Gopal sir contact number is given in the message. You all can note down those you are not having. मैंने गोपाल सर का नंबर मैसेज में डाल दिए हैं, जिनके पास नहीं है वो ले सकते हैं।
Excuse me, Vivakar sir.
Yes, ma'am.
Please add my email ID in the group, something like one.
Yeah, I have already added, madam. I have already added. No problem.
Sir, my email ID also not added.
No problem. Yours, then one person is there from Advanta Seed. Umesh sir. Umesh sir is here. I don't know whether I think he is not here. For him also problem, his mail ID is also not there. So I will add, no problem. You will get this
Gopal sir,
data
please note
sets.
on Maya number. Please note on Maya number and please add her number, mail ID also, and please
Yeah,
send
yeah.
her all details of previous session also in her mail.
You will get the data sets with interpretation files on mails.
Sir,
So no
Didakumarji,
problem.
can you add my email also?
Don't worry about the material you will get. And I will try the level best to provide whatever is available with me. So no problem. Yeah, if anyone, and one more thing, still this association mapping is not completed. We need to cover diagram wise interpretation for all the models. Again, we need to go for QTL mapping is pending. So still this association mapping will continue after the vacation.
Regarding the materials, please don't worry. After the completion of this training, you will get a printed e-manual in an e-copy to everyone.
Yeah, so that also you will get in that total scripts, example screenshot, data format screenshots, everything will be there.
You will get a
Software
single PDF.
screenshot, GUI screenshot, everything. So just you
So,
can check from that screenshot, your data file format, you can check.
is there anyone in this group who have received a call today from our office?
Yes, sir.
Yes, sir. Yes, sir. I have received.
Me also today joined, Didakumarji.
Okay. Those who have received, please reply on that number only.
Sir, I've already replied. They asked for some
Because
feedback.
from there only we are getting the list of the participants regarding the mail ID of adding. If anyone have
No,
not
for
received
me,
the
I have
call,
not received, no, I have not received call, but I have joined. Based on Sudhanshu, sir, already talked with you.
okay, those who have received, please reply on that number to add your mail, and I will get the fresh list again. Then will not face any problem. Now, if anyone have more question, you can ask. And few of the participants have given a suggestion on our comment to bring a training program on article writing. So we have decided to bring a program not only on article writing, along with that research paper writing, thesis writing, patent writing, publishing, and each and everything we are going to add so that it will be useful for you all. In this program, if anyone have any suggestion, you can drop on our number. Okay, ma'am, no problem. If you have not received the call, Kanika, ma'am, you can message me. I will look into it.
Okay, sir.
My office is 24 into 7 hours open, don't worry.
So they are easily reachable through WhatsApp and through call. So you can reach out to us if any
If you all have
difficulty.
any problem regarding accept the solution, because solution will be done by the Gopal sir regarding that topic. Other than that, if you have any problem, you can contact me 24 into 7 hours.
Okay.
Not only the daytime, even the night time you can call. I'm going to receive it. So don't worry for that.
Okay. So, sir,
Okay, so you can finish. If anyone have no more question, if anyone
yeah,
have, you can message me. No problem. I will reply to you. Okay.
yeah. So, just I will share this data sheets along with the interpretation files through your mail either by night or by tomorrow. Maximum by afternoon, you will get everything. So just do the analysis and share that to me. And then we'll meet on Friday evening. Okay?
Yes, sir. Yes, sir.
Yeah, till Friday, I will give you some work that you will receive by tomorrow. So do that work and submit to me. Okay? हाँ जी दिवाकर सर कुछ बोलना है आपको
You can close it, sir.
Okay, yeah. So guys, happy Navratri, happy Dashera in advance.
Same to you, sir.
Sir, same to you.
So we'll meet on Friday. Friday or Thursday? Friday only. Okay. So thank you.
Thank you, sir.
Yeah, we'll continue from this topic on Friday. So don't worry. Whenever I will share mail, I will reply, put a message in WhatsApp group so that you can check it. Okay. Thank you, Bibakar sir.
Thank you so much, sir.